Hello and welcome to the screencast for Chem Activity 54A, Fatty Acid Oxidation, subtitled, How is Energy Produced from Fats? This screencast is for Chemistry 120, Allied Health Chemistry at Missouri Southern State University, and I'm Dr. Michael Garut. So in this course, we are having a brief metabolism unit, and in actuality, we're not looking at any of the anabolic pathways. We're only looking at the catabolic pathways, the degradative or oxidative metabolic reactions which produce energy. We spent 3 activities looking at carbohydrate metabolism, also called cellular respiration. So if you've learned that in another class and it's been called cellular respiration, that's the same thing. And that was comprised of glycolysis, the citric acid cycle, and electron transport / oxidative phosphorylation. We're going to have one activity which looks at the catabolism or the oxidation of fats. There is an activity that has to do with protein catabolism, which we're not really going to take time for in this course, nor are we going to have a chance to do any of the anabolic reactions in this class. But at least we get the chance to do one thing past carbohydrate metabolism and see how we get energy from fats and integrate that with some other things that you're probably familiar with from the popular literature, shall we say. All right, so let's go on with Model 1. So we're going to start with fats have already entered the cell. Again, skipping a lot there. How do the fats you eat get into the cell? You can read about that in your book, but we don't have time for it. We're just going to assume, hey, the fats have gotten into the cell. And then, so this is a fat molecule, a triacylglycerol. And then the fats are broken down to release individual or free fatty acids. That's figure 2. And that's where we're going to begin. So you're going to release free fatty acids plus glycerol. That was shown back here, fatty acids plus glycerol. Glycerol is actually much closer to a carbohydrate in terms of its structure. Remember that glyceraldehyde, which is glycerol slightly oxidized, is considered the simplest carbohydrate. So when you break down a fat, you do get one molecule of glycerol, so a tiny, tiny bit of carbohydrates. But the rest of this activity is focusing on the fatty acid oxidation. And so reading the paragraph, fatty acids are activated for oxidation in the cytosol in a process that requires ATP by attaching them to acetyl-CoA to create fatty acyl-CoA molecules. And you can see that happening in figure 3. Here's a picture of a fatty acid. There's a process that requires ATP. It's creating a fatty acyl coenzyme A. This is the same coenzyme A that bonds to the acetyl group, acetyl-CoA. And an acetyl group is an acyl group. An acyl group is a generic term that means any carbonyl with any alkyl group attached to it. So the acetyl group is an acyl group. This one, which has many more carbons attached to it, is also an acyl group. And then that fatty acyl coenzyme A enters the mitochondrial matrix for oxidation, and it does that by a specific transport protein. And again, we're not examining how that takes place, except for to note that the fatty acid does have to the fat molecule gets into the cell. Once it's into the cell and the cytosol gets broken down to make the fatty acids get activated and get transported into the mitochondrial matrix. And the mitochondrial matrix is the space inside the inside the inner membrane, right? That's the matrix. All right, so now we're ready to begin. So figures 1, 2, and 3 you're going to examine while you try to answer the critical thinking questions. So let's begin with question 1. How many free fatty acids are released from each fat molecule? Pause and write your answer. That probably seems somewhat obvious. Question 2. Where does fatty acid activation occur in the cell? Pause and write your answer. Great, I talked about that, and it's also written in the paragraph right up here. Number 3. How many high-energy phosphate bonds are hydrolyzed during fatty acid activation? Explain how you can tell. Pause and write your answer. Pause now. Okay, did you say 2? There are a couple of ways you can tell. The ATP becomes AMP, so tri to mono, that's a difference of 2. And 2 phosphates, 2 inorganic phosphates are released. So remember that probably requires double the energy. Remember, ATP to ADP releases 7.3 kilocalories per mole of energy when it's hydrolyzed, and ADP to AMP releases another 7.3 kilocalories per mole. So this activation requires 2 high-energy phosphates. So when you think about this, this could be analogous to the energy input phase of glycolysis. We call it the energy investment phase, and you could imagine here that we're investing 2 ATP equivalents because it's 2 high energy phosphates. So that's the same number as for glycolysis. So now we're ready to go with Model 2, which talks about beta oxidation. This is the Greek letter beta. This is the Greek letter alpha and the Greek letter beta, lowercase. So you're going to investigate the 4 steps of beta oxidation. And let's start with critical thinking question number 4. Where in the cell does beta oxidation of fatty acids occur? Pause and write your answer. Okay, when you read the information on the first page, it talked about the mitochondrial matrix. That's where it occurs. So these are not proteins like in electron transport, which are integral membrane proteins. So they're not happening in the membrane. It's actually happening in the matrix. All right, look at the model right above and tell me which of the 4 steps are oxidation reductions. So if you want to, you can just call them steps 1, 2, 3, and 4. And then list by number which of the steps are redox reactions. And how can you tell? Pause and write your answer. Pause now. All right, so steps 1 and 3, right? And you can tell, pretty straightforward, those are the steps that involve producing those same reduced cofactors that we've seen in the past. And which other pathways that we have studied produce those reduced cofactors? So just list them here. Pause now. So certainly glycolysis produces NADH. The citric acid cycle produces both. And if you wanted to list that bridge or transition step, you could list that too. Reviewing the structures in models 1 and 2, propose a reason why this process, the process in model 2, is referred to as beta oxidation. Pause and write your answer. Pause now. All right, so it's the beta carbon that is oxidized. How do I know it's the beta carbon? That's from figure 2. For these carbonyl-containing compounds, one way of indicating the carbons is by using the Greek alphabet, beginning at the carbon next to the carbonyl and going down the chain. So I have alpha and beta, and if I were starting here, I'd have alpha and beta, and you could continue to go down the Greek alphabet, gamma, and then typically the very last one, no matter how many carbons it is usually called the omega, which looks kind of like, the lowercase omega looks kind of like a W that's really curly. omega is the last letter of the Greek alphabet, and so the omega carbon is usually the last carbon at the end. And then if you look through Model 2 and you look at the beta carbon, you see that it starts out with 2 hydrogens, but by the time you get to the end, or at least at the end of step 3, it now has an oxygen instead of 2 hydrogens. So if you have lost hydrogens and gained oxygens, both of those are shortcuts for recognizing that it's been oxidized. So we're producing these reduced cofactors or coenzymes, NADH and FADH2. What do you think will happen to those? Write a hypothesis here in number 8. Pause and write your answer. So did you say this? They'll enter the electron transport chain? They're already in the right place, yes? Because this pathway is happening in the mitochondrial matrix. If I go back to the electron transport activity and I look at the figure for electron transport, if I have NADH and I have FADH2 in the mitochondrial matrix, what's going to happen to them? They're going to go right into electron transport and be used to produce ATP. Number 9. So look back at the 4 steps. Model 2, there are 4 steps, and write the class of enzymes for each of the 4 steps. Start with number 1, and we'll do them one at a time. So do step 1. Pause now. All right, this is an easy one, right? We already said that step 1 was an oxidation. We said that because it involved the reduced cofactor FADH2, the oxidized cofactor FAD, so that would be oxidoreductase. What about step 2? So what kind of enzyme is in step 2? I will write oxidoreductase here, and then you can just go ahead and write your answer for step 2 right here. Pause and write your answer. Oh, if you are stuck, you can look back at activity 47A on the first page, which gives you the definitions and tips for identifying those types of reactions. So 47A is the place to look. All right, pause and write your answer. Pause now. Okay, so did you get lyase? Definition of a lyase: adding or removing groups involving a double bond and no ATP required. So I'm adding water. It had a double bond. I've added the OH and the H, and no ATP was required, so that's a lyase. All right, the type for step 3, pause and write your answer. Pause now. All right, you got this one. That's oxidoreductase again. And now number 4. This might be the most challenging one. See if we can give it a shot. What kind of enzyme for number 4? Pause now. It might be the easiest for you to figure this out by the process of elimination, but let me walk you through this one. It is in fact a transferase. If you look at the 2 reactants are coenzyme A and this molecule, the beta ketoacyl coenzyme A. And what you're doing is breaking it apart right here. If you look at that, this part of it becomes the acetyl-CoA. This part of it gets attached on to the coenzyme A right here, and so you are transferring this group onto the coenzyme A. So it's a transferase. You can also, like I say, do it by process of elimination because it wasn't an oxidoreductase. There were no water wasn't involved, so it couldn't be a hydrolase. There were no the double bond groups weren't necessarily involved, although that's the hardest one to exclude. Really, I think that's the hardest to exclude because there kind of is a double bond involved. This bond right here is right next to a double bonded thing. And isomerase is clearly out. ATP is not being required, so that's also out. I wouldn't give you that one on a quiz. It's a little too challenging, but the other ones I think are fair game. Let's move on. Number 10. Suppose that step 1 of beta oxidation begins with an 18-carbon fatty acyl coenzyme A, which is shown in Model 2. In that case, step 4 produces acetyl-CoA and a 16-carbon fatty acyl-CoA. And if you count them up, you can see there's 16. 1 there, 14 there, and 1 there. That's 16. And there's 17 and 18 from the original. So what do you think is going to happen to the acetyl-CoA that gets produced? Pause and write an answer. This makes sense, yes? It's already again in the mitochondrial matrix, and it can go straight into the citric acid cycle and do what we've seen acetyl-CoA does before. Remember, acetyl-CoA is that molecule in the, not this one, at the end of stage 2, it's this common molecule at the end of stage 2 of catabolism, and so it feeds into the citric acid cycle, whether it came from a fatty acid or whether it came from glucose. Part B. What do you think will happen next in order to further oxidize the 16-carbon fatty acyl-CoA? So that's this guy. Pause the video and write your hypothesis here. Pause now. So is this what your hypothesis was? I say it will re-enter beta oxidation at step 1. If I look at the beta oxidation, I have a fatty acyl coenzyme A. And isn't that what I start with? A fatty acyl coenzyme A? It's just that this one is 2 carbons shorter now. So if it's 2 carbons shorter, that leads you to have enough information to try to answer part C. Why would you say beta oxidation is said to be a spiral pathway rather than cyclic? And write that in a sentence. Pause now. So I think there are different ways you could word this, but I hope you tried to put this into a sentence. Here's my attempt. The pathway, the beta oxidation pathway, repeats, but only for a limited number of times. There is a beginning and an end. The citric acid cycle repeats over and over again because it ends with exactly the same thing that it starts with. So you could think of that like a cycle or a wheel. But the beta oxidation, when it repeats, it doesn't come back to exactly the same place. And maybe you've seen this children's toy. Was popular when I was a kid and it still exists. It's called the Slinky. It goes around, but it goes around in a spiral, right? And it eventually there's an end. So you're going to go around a certain number of times, but eventually you're going to come to the end of it because your fatty acyl coenzyme A will completely get used up at some point. Number 11. Consider the beta oxidation of lauric acid, a 12-carbon fatty acid with a similar molecular weight to glucose. So, how many spirals of beta oxidation would be needed to completely degrade lauric acid to acetyl-CoA? And answer parts A and B. Pause and write your answer to A and B. Pause now. So this is one of my favorite questions, especially part B, because when I do this in class, many times students will say 6 because it seems the obvious answer. If you have 12, well, and you want to cut it into pieces that are size of 2, well, 12 divided by 2 is 6. But you don't go through the pathway 6 times. You only go through 5 times. Here is a sentence that explains it. But I think if I draw a picture, that might be more helpful. So 12 carbon fatty acid, 2, 4, 6, 8, 10, 12. Okay. So if I've drawn it correctly with 12 carbons, then each time I go through the beta oxidation pathway, I'm going to start out with a certain number of carbons. When I finish, I will have chopped 2 carbons off, so it'll be 2 carbons shorter, 2 carbons released, and this is 2 carbons shorter. So here's my picture. There's carbon 1, carbon 2, and I cut it right here. I used a different color. All right, then carbon 1, carbon 2, cut it here. So I'm drawing a line where I've cut 2 carbon pieces off each time. 3 cuts, 4 cuts, 5 cuts. When I've cut that 5th time, the piece that's left is already only 2 carbons, so I don't have to cut the 6th time because the 2 carbon piece, which is attached to coenzyme A, what is that? It's acetyl-CoA. So that 5th time results in 2 acetyl-CoAs. So if you put that into a formula, then you could say, okay, well, it was 12 carbons divided by 2, but subtract 1 because the very last time you don't have to cut it again. So 6 minus 1 is 5. And you can do that for any number of carbons. So if you started with 18, then it would be 18 over 2 minus 1, which would be 9 minus 1, which would be 8. So you can figure this out. So 5, and we're going to call those spirals, right? Not cycles. So in those 5 spirals, answer questions C and D. How many FADH2s and how many NADHs would be produced? Pause and write your answers. Pause now. Both 5, yes? When I look at the steps, in the 4 steps, step 1 produces an FADH2, and step 3 produces an NADH, so there's 1 for each. So if there are 5 spirals, there are 5 of each. Part E. How many acetyl-CoA molecules will be produced, and what will happen to them? Pause and write your answers. Pause now. So you got 6, yes? In my picture here, I have 1, 2, 3, 4, 5, and 6 2-carbon pieces. And as those are molecules of acetyl-CoA, which are in the mitochondrial matrix, they can go right into the citric acid cycle. So now, what has been produced? Everything that's been produced can either go into the citric acid cycle or directly into electron transport. So now we're ready for part F. So, remembering that each acetyl-CoA can be oxidized in the citric acid cycle to give you 3 NADH, 1 FADH2, and 1 GTP. Right? That's good to remember. What you're going to do is do the accounting. So in the space here, total up the number of ATP that are produced from the 12-carbon fatty acid after you go all the way through electron transport, and we're using the 3 ATPs per NADH and the 2 ATPs per FADH2. So do the math here. I'll pause a couple of times and make sure that you're keeping up with me. So do the whole thing, pause, and then restart and check your work. Pause now. Okay, I'm certain you got this far. So, because we walked through it, we had 12 carbons in the fatty acid. We produced 5 FADH, 5 NADH, 5 FADH2, and 6 acetyl-CoA. So I've written that now. Now, the next thing I'll do is see what happens with these in the citric acid cycle. So I'll pause again. All right, so 6-acetyl-CoA, each of those goes into the citric acid cycle. Each of those produces 3 NADH, so 6 times 3 is 18, 1 FADH2, so 6 times 1 is 6, and 1 GTP, so 6 times 1 is 6. Now I have the total cofactors. Now let's multiply those by the appropriate ratios. So I'll pause and do that now. Pause now. So multiplying your FADHs by 2 to get 10, your 5 NADHs by 3 to get 15, 18 NADHs by 3, which is 54, the 6 FADH2s times 2, 12, and 6 GTPs, those are the same energy, remember, as ATP, so that counts as 6. So if I add those all together, I get 97 ATP. Am I done? Don't forget to subtract the number that you gave in the answer to question 3. What was that? Remember, that was the activation step that requires 2 high energy phosphates. So I'm going to subtract 2 for fatty acid activation. And I'm going to get to my final answer, which is 95 ATP. I can pretty much promise you an essay question on the quiz that will require you to do this kind of work and not for a 12 carbon fatty acid. So to show all of your work for how many, much of this comes from beta oxidation, and multiplying times all the appropriate factors to add up the ATP, subtracting the activation amount to figure out how many ATP could be produced by the complete oxidation of a particular fatty acid. You feel okay about that? Let's move on. There are a few more questions. So, compare the total number of ATP generated from one molecule of lauric acid, which is 12 carbons, to the total of 2 glucose molecules, which would also be 12 carbons. So let's write down the numbers for that. Pause now. So in the last activity, 52, we came up with 38 ATP for a glucose. So if I multiply that by 2, I get 76. And the lauric acid, which is the fatty acid, has 95. So which has more energy? If you have about equal masses of fat or carbohydrates, it's going to be fat, right? Because it produces more energy for the same approximate, the same number of carbons, right? It's actually less mass because it has a lot fewer oxygens. So, given that, why do you think organisms store excess energy as fat rather than carbohydrates? Pause and write an explanation. So I think this is the most straightforward answer. In the same amount of mass and space, you can store more energy in fat because it takes up less space and has more ATP. There are other reasons. Fats are hydrophobic. When you try to store starch, which is, let's say, glycogen, which is carbohydrate storage, it's so hydrophilic you have to have a lot of water to be able to store that. Where fats, you can just pack them right into a adipose cell, and it's pretty efficient. All right, let's move on with the last model. Ketone bodies. So this is the time in the course where we understand how low-carb diets work and whether they're advisable or not. So when your blood concentration of carbohydrate is low, such as during fasting or on a very low carbohydrate diet, or if you're diabetic because you can't metabolize glucose appropriately, in those cases, acetyl-CoA cannot attach to oxaloacetate to enter the citric acid cycle for energy. Now, let me explain that. When I look at the citric acid cycle, all of these things in the citric acid cycle, these are all carbohydrates. So they come from carbohydrates. If you don't have enough carbohydrates, that means the concentrations of all of these intermediates in the citric acid cycle is pretty low. So that includes oxaloacetate. So if you have a lot of acetyl-CoA, but you don't have a lot of oxaloacetate, then that makes this step 1 slow down, and it makes the concentrations of acetyl-CoA build up. And if they build up, then you can't get energy from them via the citric acid cycle as efficiently because they just can't get in. So when those concentrations get really high, then you have some enzymes that cause them to condense together, which is kind of the reverse of beta oxidation that we just saw where they're split apart. They condense back together, and you get this molecule called acetoacetic acid. And then there are some other enzymes that turn acetoacetic acid into these other molecules, beta-hydroxybutyric acid and acetone, and together all 3 of these molecules are referred to as the so-called ketone bodies. So let's investigate that a little bit. Number 14. So locate and circle all of the ketone groups in Figure 5. Pause and make your answer. Pause now. So there are only these 2. Remember that a ketone has to have a carbonyl and then it has to have carbons on both sides. So those are the 2 that I've circled. This is a carbonyl, but it has an oxygen on one side. These are carbonyls, but they have a sulfur on one side. This is a functional group we haven't seen before, or we haven't actually named, but it's called a thioester. Because if this were an oxygen, it would be an ester. Since I changed oxygen with a sulfur, I put in this root thio, which means sulfur. So a thioester. So the only ketones are the ones in the box there. And, you know, apparently in number 14 I said to circle it, so let's make them circles so that I can follow my own instructions. Okay, and now let's do number 15 and draw a box around all the carboxylic acid groups in Figure 5. So pause and do this now. All right, so did you find the 2 of those, the carboxylic acid groups? And what effect would high levels of carboxylic acids cause to the pH of the blood? Well, they're acids. It's going to make the blood more acidic, so the pH would decrease. Right, more acidic. So that leads us to question 16. If the concentration of ketone bodies becomes too high, as can happen in diabetics or to those on a very low carbohydrate diet, a condition called ketoacidosis can result. And you might know this condition affects the ability of the hemoglobin to carry oxygen, breathing can become difficult, it can lead to a coma, we call a diabetic coma if it's in a diabetic, could lead to death. So, considering that, why do you think this, considering what you sort of circled and boxed here, why do you suppose it's called ketoacidosis? Pause and write an answer. Pause now. So ketoacidosis, well, these are ketone bodies. So keto, they make the blood acidic, so acid, and then you have osis, which is a combining term that can be interpreted to mean a pathogenic or an abnormal condition. So there you go, ketoacidosis. Unfortunately, the term ketone bodies is kind of a misnomer, because if you look at it, only 2 of the 3 even have ketones. One of them is not a ketone at all, and really it's the acid that is more detrimental to physiology than the ketone part. So ketone bodies is just kind of, well, that's why I have it in quotes, right? Number 17. When the concentration of ketone bodies are high, acetone can diffuse out of the bloodstream into air in the lungs. Why might a physician sniff the breath of a patient on a low-carb diet to check his health? Pause and make a hypothesis. Pause now. So I'm not going to write anything down for this one. I hope you did. But notice that one of the three ketone bodies is in fact acetone. You probably know what this smells like. It smells like fingernail polish remover. Fingernail polish remover, most of that has acetone, and even if it doesn't have acetone, it has the ester ethyl acetate, which smells similar. If you smell someone's breath and it has acetone, that's pretty much an indicator that you have a high concentration of acetyl-CoA being there, and therefore not enough carbohydrates, and so that could indicate a dangerous condition. Number 18. Ketone bodies can be removed by the kidneys and excreted in the urine. Would this increase or decrease the effectiveness of a low carbohydrate diet? Explain your answer. Pause and write an answer now. So this actually does increase the effectiveness of the diet. If you're excreting ketone bodies, that's equivalent to excreting what you produce from acetyl-CoA. That's pretty much the same as saying, okay, well, if I can produce acetyl-CoA from fats or carbohydrates, and then I can convert it to these things, and then instead of getting energy out of them, I just have the kidneys filter them out and excrete them, well, that's like calories going down the drain, literally. And so that would increase the effectiveness of the diet, which is not to say that it is healthy for the kidneys. So if you go too far overboard on this, you might have kidney problems. But as far as this question goes, yeah, it would increase the effectiveness of the diet. You can lose weight this way. You just want to be monitored and be careful about it. You can sum up the major concepts in the activity and compare that with the learning goals for the activity that are posted in the Week 7 unit on Blackboard. And number 20, hey, if you have a team, well, there may not be a next time, since this is the last activity in Unit 7, and Unit 7 is the last activity in the class. How about that? As usual, you can do the exercises. These will be good practice in preparing for the quiz, comparing your answers against those posted in the exercise solutions category on Blackboard. This completes the screencast for Chem Activity 54A. Cheers!